So first speaker is going to speak on lessons of past failures. That's 30-minute slot. And lessons from past successes is a 15-minute slot. And Hilal, you explain how did that happen. But that wasn't really why I was getting up here. So I want to, yes. So there have been some recent successes overnight that will be added. So I'm very, very sad, but I'm going to steal 2 minutes of your time. Steve Finkbeiner snuck out around lunch, unfortunately, but I will highlight his work. So I guess all of you did what Sarah said, which is listen to the introductions of all the speakers. I sat at home, listened to what people do. And I noticed on my phone that at the bottom of this particular page, there was an image, and YouTube is owned by Google, right? There's an image of a person who looks like Steve Finkbeiner might look like in 30 years from now. And you might know this is Jerry Garcia, and you might know that he, just like Steve Finkbeiner, is essentially a drug testing platform, right? So, and you know that Steve talked a lot about death, and he was the lead singer in Grateful Dead. There you are. So is this a coincidence? I really thought it was a coincidence, and then I checked my own video, and I thought, gosh. And I have not, promise you, none of the stuff I'm saying now is made up. This is real. But look at this image here. My forehead. Isn't that weird? But still, we're scientists. P is not less than 0.05 yet, but it will be soon, Dan. Dan is a genius. We know that. Because if you heard his talk, I didn't understand any of the math, all this gamma, beta bands and stuff, and I thought, this is true. Dan was featured together with math genius, world's greatest math prodigy, mathematics savant. Now I think P is less than 0.05. But, Adriano, and I took these pictures last week. Yesterday, what did you say? You stood up and said, well, I don't want to start a brawl, but I'm going to start a brawl anyway because I'm Adriano. Don't want to start a brawl. What do you think you had on your page at the bottom? Larry Bird trash-talking stories that will scare you. Adriano Aguzzi. So, Google and YouTube listen to what we say, they incorporate this, and then they feature other things that might fit our interests. So with that introduction, yes. So, thank you, this is the introduction. So I'm very happy to introduce our speaker. I'm going to get rid of my sharing, one moment. Oh.
It's okay, I think I can take over.
So Ignacio is going to talk about the failures, but I stood next to you in the lunch line, and it sounded like you're going to sneak in a couple of successes in there.
Yeah.
So there'll
possibly
be failures and a little bit of success.
Okay, thanks, Hilal, and thank you to people in the NSF for inviting me. Hilal gave me a title, like everybody else, but didn't specify what I should include in the content, so I just decided to do what I wanted anyway. It was originally meant to be 10 minutes, and last night Hilal let me know that I can spend a little bit more time, so I modified a few slides. And, again, I put failures here in parentheses because, in columns, because it's not in drug discovery, a failure is, maybe defined in different ways. And I'm going to try to explain not so much, the trials that have failed to reveal an improvement in any clinical scale and therefore the drug has been approved, but rather when a particular therapeutic doesn't perform according to what was expected, how do we interpret clinical trials results to inform us as a field of the next studies? And I'm going to try to do this by comparing 3 indications: spinal muscular atrophy, or 3 monogenic indications, the SOD1 case in ALS, and Huntington's disease. Okay, I always like to start by showing a picture of the patients we're trying to help. In this case, is a woman I met in Venezuela with Huntington's disease, and just reminding everybody that there is a new generation of kids at risk for Huntington's disease that is alive and well for the time being, and that the problem we're trying to solve is important, not just for the people who are suffering the disease today, but it's important for the future generations. And I see this quest to develop therapies as really a transgenerational effort. Okay, so I'm going to start with something very basic, and apologies for some of you who know drug discovery, but there are certain things certain principles that should guide everybody doing drug discovery, but they're not always followed. The first thing is that the drug needs to engage the target. It needs to elicit a biological response and affect a clinically relevant measure. There tend to be shortcuts, either because there are methods that aren't available for a particular indication, and this is true of rare disorders, to measure each one of these steps prior to a phase 3 clinical efficacy study. And I think without the incorporation of some of these measurements, it's going to be very hard to understand the reasons for failure. I'm going to give you examples as to why that continues to be the problem. And I also want to say that today's most of the problem in neurodegeneration is a biological problem and a technological problem in the case of monogenic disorders. And I'll give examples of that too. Okay, I'm going to start with the case of SMA, and again, this is a cartoon that I got from this review, and SMA has really revolutionized, I think, the treatment of neurological disease lately, so it's an example of success, clear success. There are different modalities that are very different in nature, and I'm using this as a way of introducing the complexity of how to do drug discovery trials depending on the modality. So there are orally administered drugs, there are splicing modulators, and increase the expression of SMN2 throughout the body. There are gene therapy vectors, AAV9, SMN2, that is at the moment injected IV, although it's going to be injected intrathecally, and intrathecal injection of the ASO nusinersen. Okay, and all of those work well, but the first lesson is that when you actually administer the therapy, whether it's very early in infants or you wait longer, the functional restoration today varies a lot with the age of the subject and the time of administration, suggesting that, yes, it is a success, but it's not a curative treatment yet. And this lets us know, let us know that the timing of when you administer these therapies is going to be very significant, not only in the case of SMA, but for other indications. Okay, so now let's take the example of ALS and Huntington, and there are, in the case of Huntington, there are trials ongoing with a gene therapy, microRNA that decreases Huntington levels. There are oral drugs, so it turns out that the same drugs that have been explored in SMN2 lead to a decrease in Huntington expression through a change in splicing, and they're being pursued in phase 2 for Huntington's disease. And then there are the ASOs, particularly the Roche Ionis tominersen study that failed in phase 3, and we'll talk about this later. So first thing is there are 3 different routes of administration, 3 different modalities targeting different mechanisms with different distribution. Okay, for SMN, where the problem is mostly in the spinal cord, maybe that doesn't matter so much. In the case of Huntington's, I'll argue that it does matter because the deep nuclei of the basal ganglia need to be exposed to the therapeutic. So when we're talking about failures, we really need to understand the impact of distribution according to the different modalities. Okay, so this is the pipeline. It's a table that I use. And again, I'm just going to go quickly through this. There are antisense oligonucleotides, the Roche Ionis and the WAVE. The WAVE is allele specific. The Roche Ionis is not. Then there are programs either in the clinic in phase 2 or in preclinical development with different modalities everybody's using a different AAV, which is another complication. There are siRNAs, and there are the oral drugs. Okay, so if you look at the recent complexity in terms of Huntington splicing regulation, you probably know that there are 2 isoforms. Huntington is sufficient. The mutation only occurs in one allele. And there is reason to be concerned in the adult brain that a complete elimination of Huntington will be detrimental. So it's important to understand what of these drugs are affecting only the mutant. And the other component is that there is an exon 1 product that has been defined and produced by a missplicing event, and not all the therapies are affecting it. So In the case of the DNA-directed repressors, both of these criteria are met. But for most of the other therapies, they're not. So in terms of interpreting and comparing these clinical trial results, the modality and the molecular mechanisms matter. So the first one was stopped due to adverse events, and I will cover them. The second one, it didn't reach sufficient lowering of Huntington, and it was stopped, and all of these have stopped in development. So from a pipeline that was quite sophisticated in a period of a year, in 2021, we end up with Unicure, potentially the Roche trial is going to be reinitiated with a different dosing paradigm. Okay, so what are the potential results for failures? And again, apologies for the simplicity of this, but it's obvious, but not everybody bothers to incorporate measures that enable you to answer whether these criteria have been met. So is the dose the right dose to engage the target? And is the target being engaged in the region of the brain that you think you want to fix? And finally, there are dose limiting toxicities that can prevent the drug. For example, the splicing modulator drugs for SMN and for Huntington are quite toxic due to the mechanism. And it's unclear whether you're going to reach enough suppression in the brain to have a therapeutic effect. Okay, so measuring the target. A really important problem throughout neurodegeneration in general. We don't have a lot of assays that measure target engagement. So in the case of SMA, there are no SMN2 measures in CSF. There is no imaging. In the case of HD, we developed mutant Huntington and total Huntington measurements in CSF. And as I will tell you, we have a PET program that is now in clinical development that measures Huntington aggregates. So you can develop intracellular aggregated protein imaging tools, and I think maybe that's a lesson for what to fund. And in the case of ALS, if it's SOD, you can measure it in the CSF. The C9orf72, you have an assay for the poly-GP peptides in CSF, no imaging. Okay. In the case of Huntington's disease, I consider it a multiple systems disorder because there's so many parts of the brain that are affected. The rationale behind the Ionis ASO was we're going to try to dose very high. It's going to reach the cortex. It will penetrate somewhat into deeper structures, but that's at a really high risk given the given the pattern of degeneration that is seen in the HD case. Okay. There is very little data in terms of distribution of ASOs in humans, so this is data from the SMN trials. And again, if you look, of course, in the SMN trials is in children, so the dose and the volume administered is much lower than the two other most advanced trials with ASOs in Huntington's and SOD1, where the dose is fairly comparable. And I think these two are going to be the critical comparison in terms of safety profiles and whether they work or not. But if you look at the SMN, in a few cases, because some of the children died, you can see that even in the context of a very small spinal cord, you only get distribution of the ASOs in the spinal cord, almost nothing in the cortex, and the pharmacological effect measured just by RNA, not protein, is at the level of the spinal cord, and there is a gradient of distribution. This is probably true, if not more so, in the adult humans. And therefore, I think we need to question the modeling behind the dose selection for indications like HD, where you're trying to reach deeper structures of the brain. Okay, so that's a problem with that modality. So let me just review a little bit of the Tominer's and Huntington story. So this is the data using the assay that we developed in CSF showing that dose-dependent, time-dependent lowering of Huntington in CSF that seem to match their modeling data based on preclinical species. Everybody was very excited in the HD field. Within 2 years, the trial was halted prematurely due to safety signals that I will describe right now. And that trial has been stopped after about 18 months of the phase 3 trial from beginning to end. So this is the data from the first signal that something was wrong in the HD trial. This is the phase 2A study when they began to notice, beginning at day 85, a slight elevation at the higher doses in neurofilament and a significant ventricular volume change. Some of those individuals went onto the open label extension study, and again, they were able to see the people who switched that in the more frequent administration, but also in the less frequent one, you would see an elevation in neurofilament, okay? Neurofilament was an exploratory efficacy biomarker in the context of HD, just like it's been used in in MS or it's been used in SMN.
It was stopped for NFL elevation?
No, let me continue. That's in the phase 2. They proceeded to phase 3, but that was already an indication something was abnormal. Okay, and they said, well, there's no clinical adverse events, it's fine. Okay, well, when you actually did they proceeded to the phase 3 studies, and I'm just going to show you a couple of slides, but pretty much everything looks the same. So in this case, they adjusted the frequency of administration from 4 and 8 weeks to 8 and 16 weeks, 120 milligrams, and you can see that the people with the more frequent administration get progressively worse. The people with the once every 16 weeks, they seem to deviate in some measurements, but it's not statistically significant from the placebo control. But for the more frequent administration, everything in all the clinical HD scales, they did worse. Okay, they did worse. Neurofilament, as I showed you, increased and then came down over time, and the ventricular volume kept increasing, and it's clearly dose dependent, and to a very significant level. This is not. Some of the patients went into had hydrocephalus, and I think this is what triggered the discontinuation of the study. Okay, so what we don't know is whether this is an inflammatory response due to the ASOs, or there is a contribution of Huntington lowering in the HD population, or something unique to the HD brain that doesn't like this modality. So the comparison with the SOD1 study that is just reported on phase 3 is going to be very interesting to understand what's a modality dependent, because the chemistry and the dose are similar, versus what's a unique consequence in the context of HD. Okay. Other potential reasons for failure of the studies. Maybe you had the drug reach the concentration, but it fails to trigger a functional response. And certainly we've seen this, and I'll describe a little bit of why we think this may be the case in HD. The target may be engaged, but as the disease progresses, maybe the response to engaging that target is blunted, or there's some component of the disease that is become refractory, in this case, to the lowering of Huntington. You can have a functional response like neurofilament that works beautifully well for, spinal cord or motor pyramidal cells. May not be so informative of deeper basal ganglia structures. And finally, one thing that is certainly true in the context of HD is that none of the preclinical models have frank neurodegeneration in mice or in rodents. We tried pigs, we tried sheep, no phenotypes of neurodegeneration, and therefore everything that we're modeling is very different from the degenerating brain environment in a clinical context. So I would argue that investing in primate models of neurodegeneration is something that's very important for this area. Okay, so in my group, what we tried to do was to disprove the idea that suppressing mutant Huntington it's a good therapeutic strategy. I still believe that it is, but there are some issues that I wanted to highlight. So what we began to do is to understand what is reversible, not preventable, because most of the studies in the literature do an early intervention with a Huntington suppressing agent throughout the brain, and the animals don't develop the disease. That's great. That's not what we wanted to study. We wait for the animals to develop clear pathology at all of these different, levels, with the exception of death, which is not very common in the models that we use. And we try to understand, can you restore some of this when the animal is fully symptomatic and the phenotype is fully established? And because this was supposed to be a 10-minute talk and I don't have the data, just believe me, early intervention completely prevents aggregation. You suppress mutant Huntington, nothing forms, the cell looks very healthy. When you do this later, you can clear extranuclear aggregates. The big intranuclear inclusions remain there. We've done this for over a year in vivo with an AAV CFP. They remain. So if these are contributing in any way to pathology at the nuclear level, they're not being removed. The synaptic alterations are restored prior to and after disease onset, and this is through medium spiny neurons, subthalamic nucleus neurons, globus pallidus neurons, cortico-striatal connections. All of these things seem to be very responsive to an elimination of mutant Huntington in symptomatic animals. That fares very well for this approach. However, when you look at the transcriptional and proteomic dysregulation, when you treat early, beautiful, you get a very significant restoration or delay in the appearance. When you do it late, you go from 75% rescue to 10% rescue, okay? So there may be a refractory chromatin level event that is happening in disease progression, at least in the models, that make Huntingtin lowering not be sufficient to restore them. If this is important for clinical success, we're going to have an issue, and it requires additional intervention strategies. So some of the work that Andrew Yu is doing, I think, will be very relevant for answering some of these questions. Okay, from my perspective, one of the things that we need to do is, Huntingtin suppression leads to cellular restorative effects, circuit effects, and eventually a functional clinical improvement. What we faced at the beginning when I joined was, how do we measure this in a clinical study? And these are some of the reasons why the trials may fail, and we need to understand why they fail. So we can rule in or out that target is of valid use. So we developed Huntingtin CSF assays, mutant Huntingtin PET, PET functional response biomarkers, either in terms of synaptic density or striatal markers, and a whole bunch of other approaches that are being explored at the moment, both preclinically and clinically. So let me just tell you, we started this program from scratch. We developed a set of mutant Huntingtin-specific ligands. Initially, they were derived from amyloid imaging tools. We got away from having some activity against, say, beta-42. They do recognize ataxin aggregates. They don't recognize tau, synuclein, or amyloid pathology. And this is just an example in the mice, in non-human primates that express exon 1, and the first image of an HD patient showing weak but detectable increased binding with these ligands. We have a lot of them being explored clinically in development, and we hope that this set of tracers would enable us to understand where the therapy is having an effect if we're pursuing Huntingtin lowering. So there are other markers in the context of CSF, enkephalin, dynorphin, in the context of the medium spiny neurons. But all of this, the problem is preclinically, nobody's shown that they can be reversed with an intervention in symptomatic stages. That's work that we're trying to do. Okay. In the context of understanding the site of action, particularly for gene therapy studies, which is the basal ganglia, we started to characterize by imaging the expression of striatal markers that have been known to decrease 20 years prior to onset, both in the context of HD, in the context of dopamine receptors or PD-10. These are clearly recapitulated very early and progressively in the animal models that we use, so we use those as response biomarkers. And in order to do this, we use an allele-selective transcriptional repressor that we developed with Sangamo that doesn't touch the normal expression, but completely inhibits mutant Huntingtin expression in neurons. Well, we do these experiments, and again, those are the images with the Huntingtin tracer and PD-10, and this is a unilateral expression with the active CFP. On the other side is an inactive CFP that doesn't bind DNA, and you can clearly see the difference that gets lost over time, but is maintained one hemisphere versus the other. You can see it plotted here, the decrease in aggregation pathology, the increase or restoration of PD-10 expression. And these are some of the markers that we've tried to develop so that people can incorporate into the gene therapy trials. Okay. Another question that is important is the fact that people throw, in the case of Huntington's, a 50% suppression should be enough. And there's really no rationale why people came up with a number, other than it just sounds good and achievable. So we created a model working with Scott Zeitlin that is a regulatable mouse model where you lower Huntingtin approximately about 50% in most of the structures in the brain, both by RNA and protein. And we did an experiment where we just withdraw IPTG and therefore suppress Huntingtin. And comparing, if you just look at this initial comparison, which is 3-6 months, these are longitudinal brains, comparing the wild-type animal from the LACO Q140, you can see a very profound elimination of PD-10 signal that happens very early and is progressive. This is a wild-type animal. When you suppress mutant Huntingtin in 2 months, at 3 months and at 6 months, it's a very good preservation of that. You're pushing the disease. However, when you let those animals age, the effect is being lost. And you can see the way it's plotted here. And when you do a late intervention with 50% suppression, it doesn't get restored. So for me, at least, this argues that 50% suppression may not be sustained long enough to have the desired effect. That doesn't fare well for the clinical development. Another component that is important in the context of the gene therapy trials is the fact that CSF levels of Huntingtin, which is the only biomarker that we have today, are not responsive to an AAV intraparenchymal administration. Initially, you have a an increase due to the injury and the viral infection in NFL, which lasts a month or so, and you can see it. But in the context of measuring, this is in a transgenic mini pig, in the context of measuring Huntington levels, it's going to be unclear whether this gene therapy trial that is already ongoing in phase 2, that they have a biomarker that can tell them that it's doing what it's supposed to be doing in the brain. Another problem interpreting that trial results. So finally, I wanted to touch a little bit about the trial design or patient selection, and I'm going to use the ALS SOD trial for this example. So it is a possibility that the primary clinical endpoint wasn't appropriate for that therapeutic. The patient selection could be suboptimal. For example, fast versus slow progressors in ALS. And I will show you that this may be part of the reason the SOD trial failed. The timing of administration, as we've talked about, but how do we define how early is early and how long the trial should be? And again, the target might be wrong. You may have reached all of the things that I said. Target engagement, beautiful, but extracellular amyloid may not be relevant to clinical progression. So you've done your study well. In that sense, it's not a failure per se. The study is well designed. It's just the therapeutic strategy was not the right therapeutic strategy. Okay, so in the case of tofersen, the SOD1 ASO failed to meet the primary endpoint, which was very disappointing for everybody. Let me just show you how this study was done. So this was an SOD 100 milligrams, similar to the Huntington one, randomized 2-to-1, 28 weeks, 6 months, short study. Again, for ALS, people typically don't live very long after diagnosis, so I think they wanted to try to get a quick answer, and they thought that 6 months was long enough based on other drugs that have been approved based on a 6-month study. But then everybody was switching to the open label extension, okay? And that is still continuing. So that study, that point hasn't ended. The primary endpoint was a clinical score, and then you can see that there are key secondaries such as SOD1 levels and plasma neurofilament levels and so forth. So this is just a slide, and again, there's the link. If you have the presentations, you can click. All of these slides are publicly available. They were just presented by Tim Miller. And again, in the period of the phase 3 placebo-controlled study, which is 28 weeks, there was a trend, but it failed to meet the primary endpoint, even though it reduced SOD1 and neurofilament level. But the open label extension data is what really, I think, is making people think that the trial is working. And this is some of the data, again. So you can see effect of neurofilament is incredibly profound. Very rapid diminution of neurofilament. And the same for SOD1, I'm not showing it here. The SOD1 levels is approximately 30% in CSF. But neurofilament is clearly responding very well to the intervention. And you can see people in the placebo are slightly going up. The moment they're switched to the drug, goes down. This looks great. I mean, if this was we were seeing in HD, I'll be jumping up and down, but that's not what happens. And other measurements, respiratory function, clinical function, and time to event or time to death, all indicated that maybe here you don't have a statistical significant effect, but the moment you continue those patients for longer, they've been on the drug for longer, it appears that this drug might be working. So is it a failure or is it not a failure? When is a failure a failure? So it was a failure of the phase 3, but I think at the end this is going to succeed. And I think this is very good news for the ALS field and for the ASO community. Okay, so the timing of intervention is critical. And I think in the context of Huntington's, I just want to remind people that people have adult onset, but for 20 years we can define different levels of subclinical pathology, okay? And that is at the level of cortical volume, connectivity, alterations in CSF, alterations in imaging, whether it's structural MRI, resting state, and so forth. So we have a significant window of time where we can intervene. I think the problem is 20 years is a very long time. And how do you define people that you can follow up for long enough to afford a trial and meaningful enough that whatever market changes are happening will predict of a clinical improvement? I think that's a real challenge in chronic neurodegenerative conditions like Huntington's, Alzheimer's, or Parkinson's. And we know from the familial PS1 mutations in Colombia that that's what they try to do. They try to select a population around here that in 4 or 5 years, a lot of them will phenoconvert just based on predictions, and they realize that they were still converting into the clinical manifest phase. So just some parting thoughts for anyone who wants to get into the drug discovery space in neurodegeneration. Evaluate agent distribution and PDFX in critical circuits. I don't think it's enough to monitor CSF. for a condition where we know that you need to go into deeper brain nuclei. I think you need to develop functional biomarkers that are responsive. And again, people explore a lot of things, but they have no preclinical evidence that that biomarker responds to an intervention. Largely, that's due to the fact that we only have rodent models. And people don't want to do a PET study or an MRI study in mice. How is that going to translate to humans? I still think it's important to do this as a way of prioritizing your exploratory biomarkers. Timing, length of study, and progression landmarks. And I think here, what I see in a lot of fields is a lack of cohesion in terms of observational studies, natural history studies. Everybody's doing their own rather than coming together and say, let's define a common set of landmarks that is going to enable every single trial in terms of disease modification. And finally, I think most of the postmortem studies have been decided when somebody dies after they die. They haven't been prospectively included in the protocols. And I think this is very important, particularly in gene therapy studies, because a lot of the assumptions that we're making in distribution and the dose and the modeling for ASOs, for example, haven't been independently evaluated. And I think the data that I showed you on the SMN children really calls into question some of the model predictions that ASO companies have been putting out there. So with that, I will end, and thank you very much.
All right, we welcome questions and comments. This one in the soup?
I have
Judith.
a question. That was really great, Nacho. I wonder, for the Huntington trials, I know in HD there is a lot of emphasis on having different murine models, and I acknowledge that they are not ideal, but how well did these therapeutic approaches work in the mouse models? So obviously they must have worked well enough to move forward with human trials, but maybe with the wisdom of hindsight, if you go back to the data in the rodent models, could you have maybe predicted that they were not going to be effective or that they would have some adverse effects? I mean, if you go back and look at the data with the eyes of what you know now, could you have predicted that, and could that help you think of how to use mouse models in the future?
Yes, so the different ways to answer that question. So I don't think the mouse models are bad models. I think they're good models for certain observations that we know happen clinically, and the models are useful for certain aspects of the pathology. For example, I told you about the imaging dysregulation. The molecular dysregulation is actually quite conserved between rodents and humans based on the data that we generated so far. I think the problem is whether you're talking about a mutant-selective intervention versus suppressing both alleles of Huntington. And there's different types of paper arguing for a detrimental effect of knocking out Huntington in the adult brain by one group. Another group said there's no problem in the adult brain, the problem is in the pancreas. So we generated new conditional knockout mice. And the initial experiments seem to suggest that the thalamic calcification problems that were reported in one of the papers are reproducible. So we actually think that suppressing normal Huntington in the adult brain for an unspecified period of time in humans may not be good. So for that, I think we should have done more experiments longer term in vivo, not just in mice, but to test the idea that wild-type Huntington suppression is detrimental. The problem, I think, is with the modalities that have restricted distribution. The primate studies that were done in an intraparenchymal striatal administration only for 6 months. Okay, if you look at the neurofilament data, it begins to increase over 4 months in the humans, 4 or 5 months in the humans. I don't know if these things are connected. The ASO studies, the Unicure study is probably the most informative in that respect. But the second thing is that if you believe the mouse wild-type Huntington loss of function data, the problem is at the thalamus level, not in the striatum. So it is possible, especially with the oral drugs that we're testing, that we will uncover something simply because we just didn't do the experiments due to limitations in distribution in primates or in mice. And that's part of the problem. So hopefully that answers your question, yeah? Okay.
Could I tie into that? So the ASO trial in ALS did not see any neurofilament increases, right?
No, you saw the decrease.
Yeah, so there's a decrease. So that would suggest it is a Huntington-specific phenomenon. It's not a class effect of ASOs in general.
That's my thinking too. Or there's a problem with CSF flow or dynamics in HD that is not seen in ALS, which could be a problem. Remember also the neurofilament elevations is not immediate, right? It happens about 140 days. The ALS trial was 6 months, right? And you go very rapid decrease. So it's clear. I mean, I think this is the problem in understanding failures. For example, they didn't do any imaging on people that had hydrocephalus or people that had elevated NFL. We were chatting about this before. It's difficult to know, is the extent of lowering correlated with people that have more adverse ventricular increase or not? That intra-subject or within subject analysis has not been reported by Roche. So there's a lot of questions that we've asked them, but we don't have access to that data. So it becomes very difficult to know. But it's clearly different from the SOD1 study.
And you mentioned that maybe with the Huntington ASO, the drug or the ASOs don't really access the basal ganglia. They might mainly be in the cortex because of the route of administration.
That's correct.
So
and
would you then,
especially in the reduced dosing that they're going to do now.
Would you therefore think that the elevated NFL is related to toxicity on cortex?
Presumably, or spinal cord. It's hard to know.
And you said you hadn't access to the Roche imaging data. That's interesting. I'll have to ask about that. But so, what have you seen of the Roche imaging data?
Whatever you've seen. Whatever is presented. I haven't seen anything else.
Sounds like an exciting detective story. He's now going to figure out what I've seen.
They're keeping things very close to their chest.
I'm sure they will soon share more.
I hope so.
They will.
Ignacio, I'm a little puzzled by the really nice CSF lowering of Huntington that you saw, but yet, you know, these questions about whether you're really getting into the parenchyma of the brain. So how do you feel about is that a dichotomy or is that sort of expected?
No, the whole. Okay, so the first thing is like there's femtomolar concentrations of mutant Huntington in CSF. Okay, very small. We're playing a very small window. Number one. Number two is I show you the comparison between the clinical studies where you see this significant diminution, dose dependent. I believe that data. Okay, that's only looking at mutant. They didn't look at wild type, for example. So I don't know what's happening in wild type because we didn't have the assay at that point. But it's clearly distributing to the cortex and the spinal cord. I think most of the signal, most of the origin, I think, of Huntington CSF is probably from that rather than basal structures. The Unicure mini-pig study showed that even though you're getting 80-90% suppression in the striatum, in the basal ganglia, you only see a 13-14% diminution in CSF. So I think the CSF data may be useful for ASOs. It may not be useful for gene therapy intraparenchymal administrations, and therefore they need to find a different set of biomarkers. And at the moment, they're looking at NFL, they're looking at Huntington. So if the trial fails, we don't know necessarily why it failed.
Before you go on, say that again. In the mini-pig, 80 to 90% reduction of Huntington in the striatum, post-mortem, but in the CSF, a 13-14% decrease in Huntington.
Let me just, I think it's in, let me just go here for a second. If not, I'll look it up. But I mean, this has been published. This is not, see here, yeah. See. Why did he switch to this? Hold on, sorry.
So it's just interesting because
then. So here, okay. So this is what they showed is not in this figure. This is just the CSF concentration, right? So you can see that it goes down about 25%, but internally, in tissue punches in the same animals, you're getting 75-85% lowering. So it's not reflected in CSF to that extent.
But it wasn't that big a difference. Okay.
It wasn't that big a difference, certainly not compared to what you see with the ASOs, where you saw a very significant decrease, about 70%. So the modality, route of administration, and biomarkers need to be linked to one another.
How does Huntington get out of the neurons?
I'll tell you one thing. I think it's actually being secreted from neurons. It could be the cells are dying. So we know that the Huntington that's in CSF at least is 1,200 amino acids long, or at least there is a species that is detected with 2 antibodies, the D7F7. We don't know if it's full length yet, because the concentrations are so low. This is a hard experiment to do. We're trying to do it. But we did an experiment that we never published a long time ago, which followed some of the tau studies where people were using microdialysis. So we did microdialysis in primates and in mice, and you could detect Huntington in extracellular interstitial fluid in a normal animal. So there is some Huntington outside the cell. What is it doing there and how it gets there, that I don't know.
We had a question from Hilal.
So not sure what if these Huntington lowering therapies fail, what else is in the pipeline for Huntington?
Well, there's the concept of going after the somatic instability.
There's so much.
There's not a lot, to be honest. I mean, there are some companies that are working on some symptomatic approaches. Only one study in phase 3. The rest are very early and speculative, and the somatic instability is still very early. I think the biggest chance, hopefully, is to have displacement modulators that go everywhere. And the Unicure trial will report in December this year. So we'll see. After that, we're in deep shit, as we like to say. So we'll see.
I think the somatic instability is interesting, and there's been really good reviews written about it. So this strange phenomenon, and you correct me if I'm wrong here, but in the brain with time, the number of repeats increases to 300-400 CAG repeats. So the individual is born with 50, 60, 70, and then there seems to be a correlation between the time of onset of real frank symptoms and the emergence of these dramatic instabilities. And they're particularly in the striatum, and nobody knows why that's the case. And maybe it's related to striatum being a little bit prone to oxidative stress, so lots of DNA damage. And the other final thing is that in the GWAS that has been done to see what affects Huntington's disease progression, what pops up is DNA repair mechanisms appear. So
mismatch repair.
Yeah, mismatch repair.
Yeah.
So totally, I mean, that's really compelling. Now, what I think is interesting is that who would have imagined this 20 years ago or 25 years ago when the gene was identified?
People have speculated the somatic instability for 20 years.
22 years or something.
People didn't follow it.
Yeah.
And it's a hard thing to develop therapies for as well. So
I heard this, I should say, at a Hereditary Disease Foundation talk around 2000, 2001 in Boston. And I think it was Peggy Shelburne, who spoke about this incredible somatic instability in the R62 mouse. And everybody said, that's really weird. And everybody, including me, ignored it. But now it's
well, they're not
clear.
ignoring it now.
No.
So can I ask another question related to that? So you mentioned that, you have a kind of latency period in humans of almost 20 years. So now that you have this really nice PET dye, what happens in those 20 years? Do you not have
we don't know yet.
aggregates? Do you not have amyloids?
We don't know, Judith. So the tracer was developed as a pharmacodynamic biomarker. And it's the only thing that we could do at the time was identify small molecules with high affinity for the aggregates. We started this work 11 years ago, and it's now tested clinically. If it works well in the clinic, meaning the signal is good enough, and I think the first one won't be, but the subsequent ones will be, then we can do those studies and trying to use them as prognostic markers or essentially as a way of monitoring pathology. But it's too early to say, Judith. I think what's clear is that we need to identify, and Hilal knows about this, small molecules that bind other forms of Huntington that maybe have a higher Bmax, and therefore we can use them for these purposes. So I see this as an ongoing project rather than a single study. Yeah.
Because if you could get the neurons or like the other cells in other tissues to keep doing the same thing they are doing in those 20 years, I mean, I guess the question is there a catastrophic event or is there a gradual deterioration of? tissues, and if there isn't, right, I mean, then you could cure it just by helping neurons be resilient.
Well, my sense of this is a monotonic decrease that involves this progressive disconnection and degeneration of the cells. It's not a catastrophic event. It progresses very slowly. That's why it's so difficult to have biomarkers that are sufficiently powered to use them in a long-term study. Like if you look at neurofilament in ALS or SMN, it goes like this in a short period of time. In Huntington's, it goes like this. And therefore, to have enough of a sample size, I mean, these are going to be long-term studies with a lot of people for that type of readout. So I don't think it's a catastrophic event, and I think that cells are continuously dying. So, one of the things that the foundation is funding now is single-cell sequencing in over 100 brains, including very early stages, and we'll be able to answer the question like, is a cell sick from the beginning, and some cells are stochastically, because of somatic instability or not, becoming affected later on in disease. And it's only when the circuits break down that you start having symptoms. So all of those questions hopefully will be answered.
But I think if we don't succeed with Huntington's disease, we will not succeed with any neurodegenerative disease.
Well, you may succeed with SOD1.
No, but I think this is the simplest one of all. This is a significant population. We can access all of them. It's a monogenetic disease. There's a very long prodrome, gives us lots of time to prepare and fix it. And once the disease starts, it's rapidly progressing, so it's not one of those issues with Parkinson's with such a slow progression. In some cases, it's hard to monitor disease modification. Everything is perfect for a therapy. So we better get our act together. The thing we're missing is good imaging biomarkers. We don't have the amyloid PET type of thing. That's a weakness.
But we're trying.
And I think NFL it's not a great biomarker for.
I don't think for Huntington it's going to be, but
So that's the other
the
issue.
field has been late to this game. I think that's part of the problem.
But we better get our act together.
We're
And I
trying.
say this
We're
as a
trying.
Roche employee. All right, we're a little bit behind,
Very
but
good.
thank you so much. We're
You're
now
welcome.
going to do Jeff Kelly's going to.